Hello, my name is Hannah Nightingale, and today I will be discussing a large controversial issue in regard to the use of CRISPR-Cas9 and its potential effects of engineering on future generations. It is crucial to discuss the genetically modifying human embryos as they are the future generations to come, and the misuse of CRISPR-Cas9 can pose a large and very serious risk to society. There is a large debate surrounding the use of CRISPR-Cas9 to genetically manipulate human embryos due to the risks and ethical concerns that arise. The question to evaluate when discussing this matter is it worth the risks? First, when researchers genetically manipulate a human embryo, it can lead to the modified genes within the genetic code to become part of the environment through reproduction. This, in turn, poses a problem because if there were to be unwanted genetic changes that would not be discovered until birth, then this trait would be brought into the world, which could cause serious damage. In addition, the accuracy of CRISPR-Cas9 technology is not perfect and has not been verified to be 100%, which can cause irreversible mutations. In China, the rules and regulations differ greatly when compared to the United States. This is because China does not share the same specific restrictions or guidelines that are established within the United States in regard to genetically modifying human embryos. In fact, China has no legal basis and no detailed existing regulations established, and that is how Chinese researcher was able to genetically modify 2 twin girls to be HIV resistant in November of 2018, and when this was published, it created a very large ethical dilemma, as China does have specific regulations against in vitro human embryo modification. Scientific research has been conducted and has been used to inform the general public that the technology is not as advanced and precise as advertised. Due to the fact that CRISPR is a very easy to use and cheap technology, it can be used by almost anyone who has access to a research lab and materials. However, there is a huge misconception because it is a very long process that does take place, because the research prior to experiment or the experimental design takes years and is very expensive. However, this poses a large threat because those who are not professionally educated on how to conduct a proper research experiment on the human embryos can make mistakes, ones that could be detrimental to society and future generations. If you look to the figure to the right, it exemplifies the unpredictable and inaccurate nature surrounding CRISPR-Cas9's use on human embryos. Also, what is depicted in this image is mosaicism. However, what that is it is a condition in which cells within the same person have the genetically same makeup or identical makeup, genetic. This can affect any type of cell, whether it is blood cells, egg cells, or sperm cells. However, it is important to note the positive impact that human germline modification can have on future generations and society as a whole. For example, it has the ability to cure diseases and modify human embryos to become resistant to disease, where it will eventually become non-existent and the mortality rate of newborns and future generations will decrease significantly. Also, a reduction of the debilitating brain diseases such as Alzheimer's and Parkinson's disease that are caused by progressive cell death which can be seen by using the CRISPR technology. These diseases usually come on around elderly age, sometimes 65 and older, mainly for Alzheimer's. If you look to the image to the left, it represents the plaque growth and the entanglement of the neural connections and pathways which cause cell death. However, Alzheimer's usually affects people who have a gene called APOE, where CRISPR can be used to genetically modify the specific gene that leads to this incurable and debilitating disease. Life expectancy can also be seen to increase in future generations as a result of genetic modification of human embryos, because it can delay the aging process, which usually gives rise to cardiovascular disease as well as many others, including Alzheimer's. Lastly, extensive benefits for mental health and medical costs will provide individuals with a decrease in prescription medication and medical bills. This is because CRISPR-Cas9 can be used to modify the genome that is known to cause health issues. Additionally, it is important to review the recommendations and guidelines stated by the National Academy of Sciences, which consists of a panel of professional researchers, clinicians, and bioethicists, as they are well-educated professionals and surrounding the human genome editing. Some of the guidelines set forth and recommendations set forth by this committee state: data lacks on long-term consequences of human genome editing. Applications other than the prevention of the disease should be strictly prohibited. This includes designer babies, as an example. Public outreach and transparency is vital, and knowledge and data should be shared to the public. It is limited to monogenic diseases. And finally, preclinical evidence is required and must meet the specific criteria. Along with these, the other detailed criteria stated in the National Academy of Sciences report that was published in September of 2020 covers heritable human genome editing. The few mentioned above are only the main few that are leading. Human genome editing is a sensitive topic that has proven to raise serious ethical dilemmas. But another key factor when discussing future generations is the public's response to the idea of genetically modifying human embryos. In the figure to the right, a study was conducted in 2016 to assess whether the public saw this as a positive impact or a negative impact on future generations by basing it off of whether the adult was more worried or more enthusiastic. As you can see, the questions are as followed, or the statements. It asks the percent of US adults who say they are blank about each of these enhancements. The first one being gene editing giving babies a much reduced disease risk. 68% of the adult population were more worried about this, and 49% were enthusiastic. The second one was a brain chip implant for a much improved cognitive abilities. 69% were more worried, while 34% were more enthusiastic. Synthetic blood for much improved physical abilities. 63% were more worried, and 36% were more enthusiastic. This provides insight into what the public's opinion is on genetically modifying human embryos and humans in general. There have been experimental and therapeutic studies conducted using CRISPR-Cas9, and they have produced positive data, which supports the idea that CRISPR can decrease hereditary disease, manipulate diseases to modify their genetic sequence to show that CRISPR can be able to be used to find a cure, as well as immunotherapy, which is very important, and this can be used as a cancer alternative treatment, which decreases symptoms as opposed to chemotherapy and radiation. And immunotherapy has been chosen over chemotherapy and radiation and is very new and innovative. These technological advancements are crucial for upcoming generations, but there is an extent to which this technology can and cannot be used. In 2015, the United States Congress passed a law prohibiting the FDA from allowing germline genome editing in the United States. However, around the world, these laws and restrictions vary drastically. In the picture above, the countries in the pink have passed laws banning this modification. The United States can be seen in pink. In gray, this represents countries that have no regulations set, but it is limited to only a select few. However, the yellow represents ambiguous guidelines, but does not provide extensive details. And as mentioned earlier, in the Chinese research experimental study, which included the 2 twin girls that were born resistant to HIV, this proves that their restrictions and regulations are not clearly set. The countries in white do not have enough sufficient data to be categorized on the extent to which they have or do not have guidelines and regulations set in place. Finally, to conclude, it is evident that people across the globe understand the CRISPR-Cas9 technology in its entirety, because transparency is key when discussing human embryo modification. CRISPR is a new and innovative technological advancement that has proven beneficial across the globe. However, there are topics of great importance because the lives of generations to follow have the potential to be risked if CRISPR-Cas9 is used incorrectly with ignorance towards the rules and regulations set. As I was conducting research, a quote that came across was, CRISPR-Cas9 has the potential to either cure all diseases or kill everyone. And I think that is a very important note to end on. Thank you so much.